Taxogene is a specialized application of data harmony. It's similar in concept to the access integrity role bases that Daryl was talking about yesterday. Um, and so basically, instead of using the codes as the term names, we're using gene names. So it's a specialized application of data harmony. The thesaurus term record contains synonyms of all the different gene names that we could get. And these are the list of all the different classification schemas that we are using as synonyms. Data harmony, Taxogene was originally developed by Bob, and he worked very closely with our first customer for Taxogene, which was AACR, American Cancer
Association of Cancer Research.
Thank you. And so these are the list of all the descriptions and classification schemas that we use as synonyms for Taxogene. And right there is a screenshot. I know you guys can't see it, but it's a screenshot of our thesaurus for Taxogene, and it's kind of like, Daryl, did you show them the thesaurus, a screenshot of the thesaurus?
No, the actual.
So it's kind of a very basic flat list, which are classified in the thesaurus by a very narrow set of gene types or by RNA. And so our original client was American Association for Cancer Research. And as I mentioned before, the rule base was refined according to their content. And they had very specific content. It was biomedical research in oncology. And you can see an example here of some of their journal titles. It's very, very specific, and so it was a small matter for us when we were originally developing the rule base to refine these synonyms, these gene synonyms, to make sure that we were all talking about the same gene, because their content was very biomedical specific. And so when we originally were maintaining the rule base, they had their author submission system indexed through their subject matter thesaurus, and then they had it indexed against Taxogene. And then we would periodically index batches for them so that they could do quality assurance. They could review it and make sure that the genes that the authors submitted in their paper were actually the same genes that we were calling up. And we would do these batch indexing, give it to them for review, and then they would give us feedback, and we would refine the rules according to that. And I'm going to talk later on about when we added a second client, because we had to modify our approach to refining the rule base for this project. But with AACR, one of the indexing issues that we encountered were some gene names had different meanings according to context. So for example, we had a text to match for a gene called TF. And it's the gene for transferrin protein. But TF is also an abbreviation for transcription factor. And so when we were talking about transcription factors, here's a title and an abstract that we ran through for testing. And this up there, that's the, this is the couple of sentences with the trigger words. You can see the blue transcription factors, and then you can see in red TFs. And this was actually calling up the transferrin gene, which was not actually what they were talking about. So they were talking about transcription factors, not the transferrin gene. And so when we indexed it, this is the result that we got. We got the transferrin gene right there on the very top, which is wrong. And so our solution was to write a modification for the rule for the transferrin gene. So if MAI comes across TF and it talks about transferrin or it doesn't talk about transcription, then we're going to call this gene up. Otherwise, we're going to ignore it. And so after we implemented that tweak to the rule, it indexed properly. Another indexing issue that we came across was that the gene fusions were not indexing. And this is still a problem that we're working on. We call it the special characters project, or problem, because the reason why MAI was not recognizing the gene fusions is because they had slashes or hyphens in the gene name, sometimes colons. And so our solution was a combination of resetting data harmonies, recognition of special characters, and adding text matches of known gene fusions into the rule base. So we originally, Lamine, our lead programmer, had developed a back-end approach, a post-processing step into AACR's author submit system. And it worked for them, but it wasn't, we knew that ASCO was coming on board and it wasn't going to work for them because they didn't have the same post-processing step in their web interface. So what we did is editorially, we added text matches of known gene fusions that were, that we knew were applicable to oncology. And I'm still in the process of doing that. But so that is an example of a gene fusion. It's a fusion between gene CRTC1 and MAML2. It's separated by a slash, and when it gets indexed with test MAI, nothing comes up. And so after the implementation of our special characters fix, when we type that same character string in, we get the two appropriate genes. And so now I'm going to talk a little bit about our new client, ASCO. And they also ordered Taxogene, but Taxogene is a standalone product, and we're not specially modifying it per client as we usually do with our usual data harmony implementations for our clients. And so we had to find a solution that accurately indexed AACR content and ASCO content. And ASCO is a professional organization for oncology professionals, and their content was not as refined as AACR's, because AACR was indexing biomedical research in oncology papers through Taxogene, and ASCO had a much broader subject scope. And so some of ASCO's content includes practice management, oncology trends, and non-bench research. So it's like non-lab research, but it's still research. And so additional indexing issues that we came across was that the rules were originally tweaked with the understanding that if you came across one of these text strings, you were going to be talking about a gene. And ASCO's content is not necessarily refined to the gene environment. And so as a result, when we indexed ASCO's content, they were getting a lot of noise. And so, for example, I know nobody can read this, but this is a title and an abstract that I ran. It's ASCO's content. it's a research that was being done on survivorship care plans for oncology patients. And then they have the abbreviation of SCP. Well, that's a gene name. And so, and then if you scroll down to the bottom, you can see that word prep time. Prep is also a gene name. And then you see at the very bottom in red, hospital space in meeting accreditation requirements. Space is also a gene name. So when we indexed this, we came up with all these genes. And that paper was not talking about genes at all. And so our solution for this issue to cut down on the noise for ASCO, but still maintain the accuracy for AACR's content, was to come up with this gene wrapper. And when we say wrapper at Access Innovations, what it is, it's a programmatic add to every rule that exists in the rule base that modifies the context of when that gene or when the rule is going to kick in. And so we applied this wrapper to every rule in the rule base. This wrapper was actually developed by Gabe back there. He works very extensively with ASCO, and he knows our content way better than I do. And he's also one of our best rule writers. And so he developed this rule wrapper for the taxogene rule base, which will restrict when that rule kicks in to it being in a gene environment. And it's actually a very elegant rule, and it works very well. And Gabe's like a genius. So this rule has two parts. This is the first part, and what it does is it looks for these keywords to get you in the ballpark of talking about genetics, being in a genetics context. And I think you might be able to pick out a few words that you can see that it's like getting you into the ballpark. Then the second part is it further refines you down to you're actually talking about genetic recombination terms or other genetic researchy type terms. And so what that does is it very nicely first gets you into the ballpark and then sends you down the funnel. And so the way that this rule wrapper is designed is that you have to have one word from the first part of the gene and one word from the first part of the rule to get you into the ballpark, and then another word from the second part of the rule, which shoots you down the funnel to where we know we're talking about a gene context. And so here's a screenshot of that same survivorship care plan abstract. And now none of those genes come up because it's not talking about genes. And so the rule wrapper works very nicely to make sure that when we modify genes and we work in one rule base, that it's going to work for a very, very, very refined content area, and it's going to work for a broader oncology area.
And that also creates noise within
Yes,
that context.
that is a problem across the board in the genetics field. And with AACR, it wasn't really that big of a problem because we were embedded in their author submission system. And so when they would submit their title and abstract, first it got indexed by their subject content thesaurus, and then it got indexed against taxogene. And so our contact at AACR was a bench researcher as well, and so he and I worked together to get that content really refined and make sure that all those synonyms were there. But the problem that you described is not, it's not restricted to the genetics data sets. It's across the board when you have authors who are just submitting and they want to put in their own keywords and they just make up words not knowing that there's a controlled vocabulary for those terms. And so that's why it's really nice to have indexing at the point of author submission. Just, putting it out there that author submit is a really, really nifty feature. But thank you. Yes, Jade.
Some of those rules looked a little intimidating. Could you maybe speak a little bit to, how both programmatically when you do a wrapper kind of thing and manually, a certain amount of time, how easy or hard is it to develop rules of that level of complexity?
Okay, that's a good question. And I'm going to actually jump back and forth with describing the access integrity wrapper and the taxogen wrapper because the concepts are the same. So with both the medical data sets and with the genetic data sets, we have a lot of synonyms and acronyms that, and acronyms especially, where it can either mean a gene or it can mean it's an acronym for a medical test or it's an acronym for a disease. And we need to be able to separate those. And with the access integrity content that we were pushing through access integrity, what we were testing was actual medical records. And so sometimes we would pull up, we were finding that we were pulling up the disease name when they were actually saying that they didn't have that disease. They were ruling it out. And so what we did is we made a wrapper for access integrity's rules where all the rules would not kick in if they said these certain keywords like denies, no history, I'm sorry.
No, keep going. I have a
Okay.
question when you're finished.
So what it did is it put a wrapper in that rule was not going to turn on if they were talking about they did not have this disease or they showed no evidence of having this disease. Similarly with the rule wrapper for the taxogen, that rule wrapper defines a context first and the rule will not kick in or turn on unless those conditions are either met or not met depending on how the wrapper is designed. And so what that does is it gives us a way to refine when that rule turns on without restricting the customer's content. Does that make sense?
So I was going to ask that using or writing a rule to look for did not have sort of reminded me of the sentence. I don't remember what it's called. But
Sentence analysis.
Sentence analysis. Could you use something like that here, kind of combining the technologies to say, oh, this sentence has this word in it, but it's coming back as being more negative. Could that work?
I think that might be something that we would like to explore. But one of the things too that we do use to like kind of corral content that we're running against MAI into proper pipelines, one of the things that we do is we can search by specific fields only. And it really depends on how we build the interface and what we are pulling from the term of record. We can like parse different fields to help get us into the pipeline. And I think I see what you're asking, but that,
I have no idea.
yeah, no, that kind of is accomplished by the rule wrapper itself.
Right.
And Margie's gonna jump in.
So, I want, sorry.
You have to say AWWA when you're at AWWA, but internally we say AWA.
That's fine. I don't
It's like you don't ask for anything but tap water because that's what their standards are for there. However, they are one of our clients who most extensively uses the infield rule. The infield rule which would specify in this field, or if this field is present, Mr. Rule, will you do this instead, please? And the wrapper is a second generation infield rule design. So the path for those is fairly straightforward. Does that help? Because in your rule base particularly, you have a lot of infield rules. And Gena, could you talk about the problematic gene sequences a little bit?
Okay, so the problematic sequences and what we call the special characters problem in-house, what it is there are two gene names, maybe sometimes three, which are separated by special characters. Most of the time they're slashes. Sometimes they can be hyphens, and sometimes they're colons. And data harmony does not recognize that text string, because data harmony is, you can set data harmony to either recognize the special punctuation characters as padding characters, in which case data harmony would just ignore those punctuation marks, or you can set it to recognize it as word separators, which data harmony will see as a white space. The problem with the gene fusions and some of these pathways that we pull up when we're talking about oncology content is you're going to have a text string frequently where the special characters can be either a word separator situation or a padding character situation in the same text string. And so we understand that data harmony, we need to have a way to toggle that on and off. And according to, like, data harmony will see it as a word separator in the parsed text, but we'll see it as a padding character in the text matches, and vice versa, that we can toggle that off. And we're working on that programmatically. But that takes diving deep into the guts of data harmony. And in the meantime, we need to support our clients. And so we have been developing like multifaceted approaches to solving this problem. So some of them is that we've, as I mentioned before, Lamine had made a post-processing step in AACR's taxogene application for their Smart Submit, Author Submit system. And that worked for them, but ASCO doesn't have the same interface that we can tack that onto. So we have been working on editorially adding known gene fusions from a database that curates these, and adding them in such a way so whatever combination, whatever iteration of these two genes that are most likely to come up, you're going to get the two appropriate things. Yes, Paul.
So I'm going to stand up so you can hear me. So this is an interesting natural language processing problem. We're trying to use a character to separate words so we can identify both words, but that character also appears within words. They're not really words, right? They're text strings describing gene names. So the editorial and programmatic approaches that we've been developing are a little bit convoluted. And I wanted to say that we're working on the same problems in a slightly different approach. There's a list of this isn't my area, so you can
cell lines.
They're
contaminated cell lines.
There's a list of known contaminated cell lines. If you're a researcher who does cellular research, there are databases available online that have about 450 cell lines that are known to be contaminated, misidentified, or otherwise corrupted. Researchers are supposed to go and search this database before they submit their paper for publication. Well, 75% of researchers don't do that. So we're working on using the same technology that Gena's describing, which is leveraging MAI against a known list of, acronym medical abbreviations with all types of punctuation to identify known cell lines at the point of manuscript submission. And it involves the same kinds of problem, which is, does a hyphen separate words that you want to identify, or is a hyphen in the middle words you might want to identify? Is there slash separate words? Because the cell lines have the same abbreviation problems
Right.
that the genes do, which is that scientists like their abbreviations to spell out natural language words, right? You get extra science points if you can get the acronym for your laser lab to spell out laser. Like you get extra biology points if you can get your gene name to spell out face or if or of or these other common words. So it's an increasingly interesting natural language processing problem.
And another problem that we're finding in the cell lines, not so much in the gene taxogene rule base, but we are finding it in the cell lines project, is that a lot of researchers tend to, depending on when they went to school, they tend to want to use their old school name, or I discovered this, so I'm going to call this cell line my name, or whatever they learned that cell line name was when they were doing it, that's what they're going to call it. And there are competing attempts to standardize all these different biological classification systems, but a lot of times they don't work because, PubMed might classify something based on their subject headings, which is like what type of cell line this is, what the source is, whatever. And if this cell line is like intellectual property of University of New Mexico, they're going to have their own internal number that they prefer to call that. It's a problem in research across the board, and it's really neat how even with these challenges, we can still leverage the synonyms and data harmony and get you into the proper ballpark when we're talking about this. Yes.
Have you thought about normalizing the actual gene names or your actual terms with unique IDs?
They are normalized. So I think that we're using the main number that the term of record, the preferred term is the HUGO number, right?
Mm-hmm.
Or the HUGO name. But they're all linked with accession numbers, PubMed numbers, entree numbers, and then other, if that gene has a homolog in another, like the mouse database or the rat genome database, then it calls that one up too. So
I think you said there are about 19 synonyms per gene.
there's 16.
Possible.
Yeah, not all fields are populated though. So
You mentioned that it was a specialized version of data harmony. Does it run as a separate instance, or is it something that if you licensed it runs as within the same instance of your current data harmony? It's like just another thesaurus.
it is a specialized instance, and it's a standalone product.
Okay.
And so both AACR and ASCO have other thesauri that they have, and what they do, I don't think that ASCO is quite set up for their API call for taxogene yet, but AACR, they send a separate API call, I believe, when they, through their author submit system to taxogene. So.
Thank you.
What's all question over there?
I'm no expert, but it seems to me that I recall the way you tell the difference between genes and proteins, which have the same names, is using italics.
Yes,
And does this suss that out? And does it tell
no,
the
it didn't.
difference?
And we had a discussion with our contact Art Booker at AACR. And so we originally debated on whether we were not going to call the gene up if they were talking about the protein. But ultimately we decided that if you're in a genetics context and you see that protein and it's italicized, you still want, they're probably going to want to know that gene name. So we decided not to. We do have that in the back of our minds for in case future clients want that differentiation.
Also, by the time the text comes to us in markup language, the italics is in a tag, right? That's the only way you can identify it. And because we're processing XML documents, we have to ignore tags. If you start indexing tags, and taking them into account, then you're going to get body.
This may be a place where it doesn't matter, but later, the tags obviously have meaning.
Yeah.
And
Yeah,
can
but-
really change the meaning of
But
what
that
you're
is taken
looking
care
at.
of by the infield rule. So when we can, we could, although we are not currently, sussing out the XML fields because infield pre-processes the data it has to in order to identify.
I never thought of it that way. That's sort of
So
Yeah.
it's possible, but we're not
That is
doing it
possible.
today. To index against the tagging in the XML first and then against the rule base.
Okay, understood.